Hi, welcome to the mathematics. Today we are going to learn cell envelope. First of all, we will see definition of the cell envelope. The cell envelope is closely packed three-layered structure in which outermost glycocalyx layer, middle one is the cell wall, and innermost layer is the plasma membrane. These are the three layers present in bacterial or prokaryotic cells. They unite to form a cell envelope as a intact structure. The cell envelope is the characteristic feature of prokaryotic cells. In prokaryotes, primarily bacterial cells. Then characteristics of the cell envelope. All three layers of the cell envelope act as a single protective unit based on structural differences in the cell envelope. Bacteria can be classified as the gram-positive bacteria and the gram-negative bacteria. Then components of the cell envelope: glycocalyx, cell wall, plasma membrane. These are the three components of the cell envelope. We will see one by one. This is the generalized diagram of the bacterial cell. In this diagram, this brick red colored outermost layer is the glycocalyx layer. It might be capsule or slime layer. Then this middle one, purple colored layer is the cell wall, and innermost layer, this pale orange colored layer is the plasma membrane. These three structures unite to form a cell envelope. Then first of all, we will see glycocalyx. Etymology of word glycocalyx. The word glycocalyx is derived from two words, that is glyco means sugar and calyx means coating. This is the sugar coat. Chemically, glycocalyx is mainly composed of polysaccharide, but sometimes it may contain oligopeptides interlinked with the polysaccharide molecules. Then there are two types of glycocalyx coatings found in bacteria: slime layer and capsule. The first one, slime layer, is the weakly bonded with the cell wall and looks like a slippery or viscous layer. Slime layer in bacterial cells performs very important functions like adhesion to any surface, which is facilitated by the slime layer. Then slime layer prevents loss of water, it prevents loss of nutrients and protects bacterial cells against viral attacks. Then capsule. Capsule is the thick, tough and strongly bonded with the cell wall. Functions of the capsule are like adhesion, protection against phagocytosis, and protection against some type of antibiotics. Then cell wall. Cell wall is the thick, rigid middle layer of the cell envelope. Then composition of cell wall. Chemically, cell wall is composed of peptidoglycan, that is murin or mucopeptides. Glycan part of the peptidoglycan is composed of two types of polymers, that is NAG and NAM. NAG means N-acetyl glucosamine and NAM means N-acetyl muramic acid. Functions of the cell wall. In bacterial cells, the cell wall determines shape of the bacteria and provides strong structural support to prevent bursting or collapsing of the bacteria. Then chemical and molecular variations that are present in bacterial cell wall differentiate bacterial cells into two categories, that is gram positive and gram negative. The bacterial cells that take up a gram stain are recognized as gram positive, and bacterial cells that do not take gram stain are recognized as gram negative bacterial cells. The difference between gram positive and gram negative bacteria: gram positive bacteria stay blue or violet colored with the treatment of gram stain, that is crystal violet, even after treatment of absolute alcohol or the acetone. Whereas gram negative bacteria are unable to retain gram stain after the treatment of the absolute alcohol or the acetone. In gram negative bacteria, the outermost layer, the cell wall, is the lipopolysaccharide layer that dissolves after the treatment of alcohol or acetone, and gram stain gets washed out. This is the reason why gram negative bacteria do not show or do not bear gram stain. Then gram positive bacteria show single layered cell wall, and gram negative bacteria show double layered or the two layered cell wall. In gram positive bacteria, the outer membrane is absent. In gram negative bacteria, the outer membrane of lipopolysaccharide is present. The thickness of cell wall in gram positive bacteria ranges in between 20 to 80 nanometer, and in gram negative bacteria, thickness of cell wall ranges in between 8 to 12 nanometer. Gram positive bacteria show thick peptidoglycan layer with teichoic acid embedded inside, whereas gram negative bacteria show thin peptidoglycan layer but there is no teichoic acid embedded inside it. In gram positive bacteria, mostly capsule is absent. In gram negative bacteria, mostly capsule is present. Cell envelope. This is the diagram, generalized diagram indicating cell envelope present in the gram positive bacteria. In gram positive bacteria, this is the plasma membrane, and this is the peptidoglycan layer, whereas this is the generalized structure of cell envelope present in the gram negative bacteria. In gram negative bacteria, the outermost layer or the outer layer of the cell wall is the lipopolysaccharide layer. This is the lipopolysaccharide layer, and this is peptidoglycan layer and plasma membrane. This is the plasma membrane, and this is periplasmic space. This periplasmic space contains various enzymes, and these enzymes are responsible for the sustainability of the bacterial cell against harsh environments. Plasma membrane, then last layer. Plasma membrane is the innermost layer of the cell wall, and plasma membrane is semi-permeable type of layer. Functions of the plasma membrane. Plasma membrane performs functions like nutrient transport. Then plasma membrane provides site for the respiration and photosynthesis. And in bacterial cells, plasma membrane is the site for the synthesis of components of the cell wall. These are the references. Thank you for watching. Please do subscribe and hit the bell icon. Thank you.